Thank you, Professor Saptarshi, for that very kind introduction. And thank you to Professor Bandyopadhyay as well for inviting me here, for chairing the session, and for inviting me to give this talk. So, like Professor Saptarshi mentioned, we have a couple of different themes in our lab, all centered around what we like to think of as polymeric biomaterials and pharmaceutical soft matter. And in today's talk, given the theme of the track in the session, what I thought I would do is focus more on the pharma part of the lab, a little bit talk about that, and then talk about how we use drug functionalized electrospun carriers for biomedical applications. I want to start out by giving a quick overview of what our lab does in general before I move to um, the specific talk for the day. So, like I said, our lab, is there any way I can hide this on top? Broadly, like I said, our lab is interested in processing of polymeric biomaterials and pharmaceutical soft matter. And one of the bread and butter of our lab has been this process of electrospinning, which I'm going to talk about in just a little bit. But very briefly, it's a process to make process polymeric solutions and make fibrous meshes. And on the processing aspect, we're more interested in understanding how polymer solutions interact with electric fields and how the charge plays a big role in forming fibers, right from the application of charge all the way to the point where the fibers get collected. And from an application perspective, we're interested in developing maps such as these that can inform, industry personnel, for example, about stable modes of operation for getting fibrous meshes with specific properties. Um, Using the insights gained, mechanistic insights gained from these experiments, what we do is we're interested in nanofibrous mesh design. And here we're interested in rational design of functional meshes, particularly for pharma applications, where we're looking at functionalizing these materials, developing technologies and platforms to create materials that can give zero order release kinetics, for example, some of which I'm going to talk about today. And from here, we can go into two different directions. One direction is development of amorphous drug formulations, where we develop property function relationships, where we've developed technologies again to, I'm going to talk about that today a little bit, to develop materials that can give release kinetics that match with predicted release kinetics. More recently, we have gotten into co-amorphous drug systems. These are more for oral solids. Again, this is in collaboration with NIMS Japan. I'm not going to talk about that today. And on the more biological aspects, we're equally interested in deploying these materials in biomedical applications. And here our interest has, for a long time, has been addressing the problem of chronic inflammation, which I thought would be of interest to this audience. So in the interest of time and given the audience, my talk today will focus broadly on nanofibrous mesh design with insights into how we can design materials for achieving specific release kinetics of small molecule drugs. And then the second part of the talk, I'll talk a little bit about how we use these materials for controlling chronic inflammation in specific retinal vascular diseases. So that being said, I'll give a quick overview of electrospinning in case some of you are not familiar with the topic. So this process is a very, very old, 150-year-old process, perhaps. For example, Kelvin has talked about some preliminary aspects of this process. So that should tell you how old the process is. So essentially, it's a process where a polymeric solution is taken and is pumped through a metal orifice, like a needle. And as it comes out, a very, very high electric potential is applied. And as the electric potential as the field strength keeps going up from left to right, you initially have a droplet that's suspended because of surface tension. And as more and more charges get pumped into the solution, electrostatic repulsions deform the droplet into a cone, which then, because of excessive charge, starts to eject the jet, which then stretches over and thins over several orders of magnitude to give what we call nanofibers. And going just to give you an idea of the order of magnitude reduction we're talking about a millimeter here, for example, all the way up to a micron or even lower on the nanofiber diameter. Electrospinning is a fairly complex process, and there are several variables that are involved in controlling the properties of the fibers. Now, why are we interested in controlling the properties of the fibers? Because those properties such as diameter and porosity and pore size have a direct impact on how these meshes are employed in biomedical applications. So, and my lab is quite interested in understanding how these different electrospinning conditions and solution variables affect the properties of the fibers, which again, I'm not going to focus on much today. But like I said, the key takeaway is all of these properties of the fibers are affected by solution electrospinning parameters. And in turn, these affect how we employ these materials in biomedical applications, particularly control release applications. The meshes that we make are largely suitable for biomedical applications because we're targeting a fiber diameter in the range that is known to influence cell responses, and some of which I'll talk about today. Electrospun meshes, because they're non-woven fibrous meshes, they are architectural mimics of the extracellular matrix. So cells can naturally attach to them and grow. And because of the large surface to volume ratio, they also permit very, very high drug loading. Right. So with that quick introduction about electrospinning, let me jump right into a story that we began some years ago. So we have largely been interested in looking at chronic inflammation from an application standpoint. And the drugs thus are tailored towards looking at corticosteroids and the like to see if we can load some of these drugs into electrospun fibers and then release them over long time periods to control chronic inflammation. So one drug that I'm showing here is this dexamethasone. And this is a story that we put out some years ago. And I'm not going to talk too much about the story particularly, but I want to use this as a prelude to talk about the next set of steps. So here in this work, what we did was first try to understand and come up with a framework for understanding how matrix properties such as mesh architecture, polymer chemistry, mesh degradation, drug polymer interactions, etc., lead to differential release kinetics. So we took five different or four different polyalpha esters, which are biodegradable and clinically approved for such applications. And then we put dexamethasone into all of these fibrous meshes. And then what I'm showing you here is the cumulative release kinetics. And what you will immediately see is that we get a wide spectrum of release kinetics going from almost no drug release from one of these materials to sort of zero order kind of release kinetics to very, very quick burst release. So in this work, we went into the details of understanding how these drug release mechanisms are guided by many of these properties. Now, I wanted to keep this picture in mind because I'm going to use this to talk about the story that I'm going to talk about today, which is trying to now focus on two of these aspects. So from this, we're going to look at this material here that shows burst release. And one of the questions has been, can we control the burst release? Second one is looking at this material that shows sort of zero order release kinetics. And so why is this important? Many of these drugs that are out there have very, very low solubility and have very weak interactions. So typically when you put these drugs in a matrix and try to deliver them, what happens is you get what is called a burst effect. Essentially what will happen is a lot of the drug will come out initially for whatever reason. This could be driven by poor drug polymer compatibility, phase separation, poor drug solubility, a bunch of factors, right? But nevertheless, the burst effect largely is where the drug comes out very, very quickly. And that's something you want to avoid because that can have a lot of adverse effects on the application and also can give very, very low long-term efficacy because most of the drug comes out in the first few hours or few days. So the zero order paradigm is an interesting and a very lofty paradigm in the drug release community, where you're looking largely at a release rate that remains constant over a time period. That's independent of how much drug you put in. The amount of drug released over time remains the same. Now, many people have done it using many different approaches. Our key aspects in this study that I'm going to talk about are two things. One is we didn't want to use additional excipients or we didn't want to have barrier layers. For example, you can have two barriers in between the two meshes and slow down the diffusion. That's one way to do it. You can change the release kinetics using cross-linking. You can do it using the addition of excipients. So we were at the outset wanting not to go with those kinds of approaches. They have their advantages, but they also have limitations. The second thing is we also wanted to achieve zero order release kinetics for a very, very long time periods. And when I say long time periods, I'm talking about days, several days to weeks, because we are targeting these as parenteral applications and not oral solids. So with that in mind, the question we asked is, how does one achieve tunable zero order release rates without significant burst effects? And here, guided by our experience with the dexamethasone that I showed you on the previous slide, we took another corticosteroid budesonide and we picked those two materials that gave us very, very significant burst release and zero order kind of release over several days. Now, what we've done here is we've just taken the drug budesonide, put them into two different materials. One is polycaprolactone PCl, the other is polylactide glycolide, which is PLGA, and we've shown you the release kinetics here. The question now is, these are intrinsically driven by how the drug interacts with the polymer. Now the question is, can we achieve tunable zero order release rates? By that I mean, if you wanted the drug to release at a faster rate from this material, but still retain the zero order kind of profile, or can you reduce this burst that you see with PCL? How do we achieve this? So with that in mind, the first thing that we thought we would look at is a system-based approach where you're looking at rationally guided blends. So we have a polymer that releases slowly, a polymer that releases fast. First step is, can you look at blends? And blends are intrinsically problematic, as I'll show you, because from a thermodynamic perspective, they are very unstable. You will get a lot of phase separation. So we first went with 20% PCl and 80% PLGA. And shown here is a bar graph of the composition, showing that we indeed have what we are saying we have in the mesh. So these are just blends of these two, the pink and the black curves. And you see that having more of the PLGA as compared to less of the PCl, you sort of go from here to here, or from here to here, depending on which way you want to look at it, right? But then the next step is, can this be more systematized? Is the next question we asked. But as you quickly see, when we go from 20% to 30% PCl, we immediately get back this very, very sharp burst release. And then we asked, is it always true that you're going to get this? So we went much higher with PCl, and that again shows the same burst release. So what we were able to conclude from this is greater than 30% PCl will induce significant burst release. And in the interest of time, I'm not going to get into the details of why this is happening in great detail. I mean, the reference is provided here. But I will talk a little bit about the mechanisms involved here. So we were looking at a bunch of different solid state characterization techniques, right from DSC to IR spectroscopy to Hansen solubility predictions. And what we were able to show largely is that one of the reasons why, a couple of reasons why PCl shows this burst release. One is the TG is very, very low. So at room temperature, the free volume is much higher because the polymer is no longer glassy, it's very rubbery. So the drug, it's able to diffuse out very quickly. Second thing is the Hansen predictions also show poor miscibility between drug and polymer for PCl. So anything above 7.5 is considered partially miscible, and anything less than 4 or so is considered miscible. So you see that for PLGA the drug is fully miscible in the solid state, whereas in the PCl the drug only shows partial miscibility. That also meant that we were able to look at water uptake ratios for these meshes, and we were able to show that there was a strong correlation between these are the Pearson correlation coefficients, a strong correlation between the water uptake ratio for these blends, 30-70 blend, and the release rate. And the point here I'm making is that because the drug is incompatible with PCl, it phase separates within the PCl matrix. And because it phase separates, the water is able to penetrate, whatever water penetrates on the surface, the drug comes out very quickly. We were also able to show using DSC that this is also guided by differential drug polymer interactions between the PCl drug and polymer and the PLGA drug. So the factors to summarize that slide here is that we're looking at glass transition temperatures, differences in water uptake, polymer visibility, and drug polymer interactions. So this is a good starting point. We're able to reduce the burst using blends, but this is still not good enough because we're still, it cannot be systematized in the sense that I cannot scale it up. When I go to higher blend ratios, I'm still getting back the burst. So to look at trying to control or mitigate the burst from those kinds of systems, we developed, this is something that we pioneered in our lab, which is to look at the simultaneous co-electrospinning, where we have two different fibers integrated simultaneously into a system. So what's happening here is instead of a single spinneret delivering one polymer solution with drug, you have two spinnerets delivering two different polymer solutions with the drug rationally distributed across the two fibers. Because the fibers are independently integrated, you can decouple the properties. And because they are not blended together, visibility and phase separation are no longer concerns. And if you can intelligently pick how the drug is distributed across the fibers, you can get predictable release kinetics. So first we showed that we indeed have these two sets of fibers. So shown on the left is a bright field image of two sets of fibers with two different dyes in them. And fluorescence image shows that indeed we have two different sets of fibers co-integrated using the simultaneous electrospinning process into the same matrix. But the real question is, does this actually give you the predictable release kinetics that you think it will give you? So the first thing we show is that between, so I'll walk through this slide. So what we're looking at is, if you remember, I showed you that the 30% blend shows burst release. So what we wanted to do is, the question we asked is, can we keep the PCL PLG ratio the same and still get lower burst release? So what we did is, we co-electrospun such that we get the PCL PLG ratio to be the same in the co-electrospun sample. So this plot here shows that the compositions have not changed, except in one it is a blend, in the other it's a co-electrospun sample. But we show that because of co-electrospinning and because of the rationally guided distribution of the drug across the two sets of fibers, from this blue curve to this green curve, which cannot be achieved using just blends alone, right? And because these are co-electrospun with the drug distributed exactly the way we wanted, we're able to show that in the co-electrospun sample, we're able to get drug release that matches predicted release profiles, whereas in the blends, they just diverge as time progresses and making it worse and worse. That's largely due to poor visibility and unequal drug partitioning, whereas in the co-electrospun sample, because of precisely engineered drug distribution, we get predictable release. And of course, we have fit them into different zero order and first order models, which details of which again are available in the work. So 2 points I want to highlight here is, between the blue and the green curves, the composition of the matrix remains the same, overall composition. What we are able to achieve using this co-electrospinning process is decoupling of fiber properties, and thus being able to release drugs using this matrix in a predictable manner. Second thing is, we're also able to achieve this kind of profiles over many, many days. So it's not just a few days, a few hours, we're able to achieve this over many, many days. Currently, the work we're looking at moving forward is development of co-amorphous drug formulations, where again, I'm not going to talk about that much today, but idea here is, can you develop matrices where there are two drugs in the co-morphized together, two drugs co-morphized together in the same matrix, to look at release profiles of multiple drugs from the same matrix for combination therapy. So in the remaining time that I have, what I want to do is switch gears a little bit and then ask a broad question and try to answer that in the remaining time that I have, which is, we've shown our ability to load drugs, corticosteroids, into electrospun matrices, and we've been able to show that we can control the release profiles. Now, what are the effects of these things in a biological setting? So if you actually deploy these materials, for example, electrospun materials with or without drug, in a condition where there is chronic inflammation, which is primarily what these drugs target, what are the effect that they have? So this work is done in collaboration with Dr. Indrajit Kaur at LV Prasad Eye Hospital. And the problem we are looking at here is chronic inflammation, but in the context of retinal vascular diseases. Now, these are conditions that are widely prevalent in countries like India, for example, where diabetic retinopathy and retinopathy of prematurity result in damaged retina and vision impairment. So, for example, diabetic retinopathy, because of the underlying diabetic condition and hyperglycemia, chronic inflammation persists. And as a result, the retina starts to get damaged over time. This is what I like to call the unholy triad, if you will, where you have chronic inflammation, you have hypoxic or hyperglycemic stress. So hypoxic is just lack of oxygen deprivation. Hyperglycemic stress is excessive blood glucose levels. So these two cause a significant amount of chronic inflammation in the body. And the key cell type involved here that we're targeting is a cell type called the microglia, which is an immune cell. Broadly, it's known to exist in multiple states, but here we're looking at two different states where you have these cells can take up a pro-inflammatory state or take up an anti-inflammatory state. Broadly, I mean, these are very broad paradigms. There's a lot of subtext there, which I'm not getting into. But for the purposes of this talk, we'll simply have to go with the idea that these cells can broadly exist in two different states. So one of the questions we ask is, because the microglial dysfunction is involved in diabetic retinopathy, for example, it is well known that cells constantly remain in this pro-inflammatory state in this disease. Can we now deploy electrospun materials along either on their own or functionalized with drug to target these microglia, to switch them to go from an M1 pro-inflammatory phenotype to an M2 anti-inflammatory phenotype, with a view to controlling chronic inflammation in the long term? So what we first did is, here we started off by saying, let's start with the material alone and not put the drug in first. So here we were looking at in this work, again, I'm giving the references so that in case somebody wants to look it up, can always look it up. In this work, we were trying to look at, can the materials on their own control microglial responses under hypoxic stress? So based on previous findings in the literature, we were able to use matrix architecture as a cue to control cell shape. And here we prepared two different sets of materials. One was randomly oriented, the other was aligned matrices. And in biology, one of the paradigms broadly is cell shape controls cell phenotype. So, if you go invert that argument, can you control cell shape by engineering materials with different architectures and hence control cell phenotype? So what we are able to first show is by controlling the fiber orientation in the electrospun mesh, and this is without drug, we were able to control the cell shape. These are microglial cells, CHME3 microglial cells. So we were able to show that nanofiber orientation guides cell orientance and cell shape via contact guidance, right? But the key question is, what do these differences translate to in terms of phenotypic differences under hypoxic conditions, which is known to persist in diabetic retinopathy? So there we were able to show using gene expression and protein expression studies that when you have these random and aligned meshes, so the black and the gray bars are normoxia, and the blue and the red bars are hypoxia. And what we were able to show is under hypoxia, we were able to show that not only do these meshes on their own intrinsically suppress pro-inflammatory markers, they also significantly upregulate anti-inflammatory markers. One way to visualize that in the immune cell community is to look at the M2 to M1 ratio. So for example, here IL-4 is a cytokine that's associated with the M2 anti-inflammatory phenotype. IL-6 is a cytokine associated with the M1 pro-inflammatory phenotype. So not only are we showing that the pro-inflammatory cytokines are suppressed and anti-inflammatory cytokines are upregulated in the presence of meshes under hypoxic stress, we're also able to show that the M2 M1 balance is now completely switched in favor of M2 cells, which are the anti-inflammatory phenotype. And we were also able to look at genes associated with cell attachment and cell shape. And we were able to show that these meshes beneficially alter cell function to a repetitive phenotype through an attachment-driven mechanism. So having established the intrinsic role of the meshes in modulating cell phenotype under hypoxic stress, the next thing we looked at was, what if we load a corticosteroid into these meshes and then look at those effects under hyperglycemic stress, which is the other trigger in diabetic retinopathy. So for that, we're looking at dexamethasone, which is the drug that I talked about earlier, loading dexamethasone into electrospun meshes and trying to target chronic inflammation, now under hyperglycemic stress. So what we first did is, using our understanding of drug polymer interactions and whatever I talked about in terms of our knowledge that we've developed over the years in understanding how drugs release from polymers, we were able to load dexamethasone within the PLGA fibers. And here we knew we wanted control release over a very long time period. So we went with PLGA that has previously been shown to give zero order, as I showed you. So first we confirmed using IR spectroscopy that indeed the drug is present within the fiber. You don't see that peak in the black line, which is just PLGA. But what we're also able to do is advance our understanding of drug polymer interactions a bit more. We've done Raman line scanning here, and I'll walk you through this a little bit. So shown here is a Raman spectrum, and the red and the green are peaks marked for the drug and the fiber and the drug respectively. And if you take a single fiber, as shown here, and the arrow points to where we have done the Raman line scan. So if you go through the fiber radially, and you plot the normalized intensity of the drug and the polymer, what you were able to show is the drug is uniformly distributed. And because of the differences in intensities, we're showing a inset here for the drug peak alone. So what you're seeing here is from left to right, I'm going through the fiber. And we were able to show that as you go from left to right, you get uniform distribution of drug throughout the fiber, which indicates good solid state miscibility of the drug with the fiber. And this is in line with the expectation based on our understanding of how these drugs interact with PLGA. That in turn translates to
uniform, or like, is it maximum at the center?
It is maximum at the center, so that there is a maxima that it goes through. What I've not shown you is for PCL, where you will see it mostly on this surface. which leads to that burst release that I talked about earlier. Again, I didn't include that in the interest of time, but that's what you will see. So in turn, we were able to show that because we were able to do this, we were able to achieve this kind of release of the dexamethasone over not hours, not days, but over weeks. So this is over 8 weeks because we're looking at something that can be injected or implanted in the eye, and you can't do that every week. So, and you notice that over 8 weeks, we still not even released 50% of the drug. And again, this is my last slide. So we then ask, can this drug and mesh have an effect on the cells? So again, we show that as compared to the, so CS is just last cover slip, as that's a control. As compared to the PLGA alone, when you have the DEX loaded into the PLGA, we see that markers for both MMP9 and IL-6, which are both pro-inflammatory markers, are suppressed. DEX and fiber together also promote higher cell viability under hyperglycemic stress. So HG is hyperglycemia. And we were also able to show using protein expression studies that as with the meshes alone, DEX with meshes synergistically promote a decrease in pro-inflammatory markers and an increase in anti-inflammatory markers. So what we're now looking at is simultaneous release of multiple drugs for combination therapy, targeting not just inflammation, but other conditions associated with retinal vascular diseases. So in summary, I hope I've been able to convince you of some of the things that we've been able to do in terms of looking at drug polymer interactions and loading drugs into electrospun fibers and releasing them at controlled release rates. And also in our expertise in looking at those kinds of materials and being deployed in targeting chronic inflammation in biomedical applications. So with that, I want to thank my lab. So the work you saw today primarily was done by Nikita, my first PhD student, and Divya, my one of the PhD students who's about to graduate now, and many, many others who contributed to it. I'm very thankful to the funding agencies for funding our work. And I want to thank the organizers for inviting me here. I'm happy to answer any questions.